All right, so it's noon, so we're gonna get started. Hello, everyone. Welcome to today's webinar. My name is Rosa Rodales, and I will be the moderator for today's session. We ask that you please remain muted during the presentation and save your questions until the end of the presentation. You can type them in the chat box or you can raise your hand and you will be unmuted at the end of the presentation. Today's webinar is the third of a three-part series of webinars that was sponsored by the Northeast Greenhouse Conference and its Growers Association. And our speaker today is Margery Daughtrey. She doesn't need introduction, but I'll just say something briefly. She is a senior extension associate at Cornell University, and Margery does extensive work improving disease management for ornamental crops with the emphasis of developing and communicating new information for the benefit of the industry. So she has helped a lot of us out there. So very glad to have her today with us. So with that, take it over, Margery. And thanks for being here.
Thank you so much, Rosa. It's nice to be here. I have friends in the audience, I know, and I'm happy to be able to talk to you about diseases. And I was able to choose them at my discretion because of the title. We'll be going into diseases caused by fungi, bacteria, nematodes, and viruses. So there'll be something for everybody, I hope. And you probably are familiar now with the different pathogen categories. So hopefully you noticed there was something missing when you saw that list. The question is, what else could I be talking about that I'm not going to be? And that's actually the water molds. If you're a Pythium or Phytophthora fan, you won't be hearing about them today. You also won't be hearing about downy mildews today, just so that I can fit it all into one session. But maybe we'll do that in another year. Because we certainly don't want to ignore the diseases caused by the oomycetes. So let's start out about thinking about the fungi. And when you're thinking about controlling fungal diseases, you really need to think about water and its management, and also think about good sanitation. This happens to be salt water, but it's a good water illustration. So we'll use that as our backdrop here. And we'll talk about just 4 of the fungal diseases that are important to you as a grower in the northeastern part of the United States. And we'll go through the black root rot, which we used to call Thielaviopsis, but is now Berkeleomyces. We'll talk some about powdery mildews and some about Fusarium diseases and some about Cercospora. And let's start off with the old Thielaviopsis basicula that's now Berkeleomyces basicula, which causes a disease with a much easier name to say called black root rot. And first of all, you need to know where it's going to show up. This is not a disease that gets all of your crops, but it does get more than one. And it's important to understand that it might move from one crop to another if you have more than one susceptible crop in your mix. Calibrachoa is the crop where we've seen the most problems in recent years. It was a new crop at one point. And although we'd had the possibility of petunia infection, it was interesting how much Calibrachoas started getting it. Perennials may show the disease if you're a perennial grower. Poinsettia had a classic problem back when we used to grow the plants in soil rather than a soilless mix. Vincas and violas and pansies are very prone to the disease. I've seen it on rosemary. And there's this curious thing about getting it on basil if you're growing them hydroponically. And if you're a vegetable grower in the South, you've probably seen the disease there as well. One of the recent samples was on a dianthus, and it shows some of the symptoms that you might run into with the black root rot. Overall, there's a tendency towards stunting because the root system isn't very functional. And there's often some yellowing, particularly of the lower foliage, and some discoloration could be purple instead, just showing a lack of nitrogen getting up to the plant. And it's going to look sickly compared to others. The close-up, if you're looking at the root systems, includes the ability to look directly at the spore production of the fungus. Those funny little things there that look a little bit like Rolos or Tootsie Rolls are the structures that this fungus makes to survive long periods without a host. And because they're dark colored, they survive even a little bit of exposure to UV light, and they're less likely to be broken down by other microorganisms. So this is what's lingering in your pots and in your flats. If you have the disease in one season and then happen to see it come back again the next season, that's why we really stress that you need to clean up any of the pots or flats or just throw them away altogether if you have a year where this disease shows its head. Here's a better illustration of how the fungus operates. You've actually got a couple of spore types. You've got that resting spore, that brown one, which is designed for long-term survival, but it also makes things called endospores, which are squirted out of these little tubes. Here's part of an endospore, here's part of a sclerotium here, and then there's this strange little projection, and these little rectangular type spores, or they're actually more like Vienna sausages, I should think in 3 dimensions, get squirted out of these cases, and then you have them able in particular to move with water. They'll float easily from place to place. It's a soil-borne fungus. It will move with soil, but it can also flow with water and get distributed in the greenhouse. When you bring in plants of a susceptible type, look at the root systems. You want a root system to be white and vigorous looking, like the one on the left with the blue background there at the bottom right. That's the kind of root system you want to plant. If you see lack of roots, or if you see blackened or dark brown roots, that might be an indication that you've bought some Thielaviopsis along with your liners. Here's a close-up of a plant in a larger pot, and you can see some white roots here, but they're large patches that have that very dark brown look that comes with the Thielaviopsis. This is one that I inoculated, so I understand why it looks sick, but you need to question why your root systems don't look perfect in your own greenhouse operation. As you're growing plants on, you'll know that you have Thielaviopsis because the plants will be growing much more slowly than a healthy plant. And the general situation is that you won't have all of them sick at once. So there will tend to be a very irregular stand of plants. You might have one pot where plants look healthy, one pot where they look very stunted and yellowish or purplish. If you are growing hydroponic basil, you will have perfectly healthy roots at the top of the cube, and then down at the bottom, they'll be extremely dark and discolored and softened because they have the Thielaviopsis disease. And that black root rot is going to be coming in more aggressively on the lower part of the root system because it thrives in wet conditions. And this is a lesson to you not to have overly wet conditions in your regular culture as well. That will actually favor the Thielaviopsis. Here's the large number of spores that we get when we culture on carrot slices. One of the best ways to identify this disease in a laboratory is actually to take a piece of root that you suspect of having the disease and lay it across a carrot slice. It just loves carrots. So you'll get huge amounts of sporulation that you can easily identify. Here's an example of that irregular stand look. These are Shasta daisies, with some plants more stunted than others and discolored. And when you see plants that aren't healthy like this, you need to examine the root system. That's where your answers are going to come from. The environment that favors black root rot is a high pH, and that's just natural to some of the places where you live. You'll need to fight that if that's your natural pH and apply some sort of acidification to get your mix so that it doesn't favor the disease for the susceptible crops. The wet soils, as I mentioned, large numbers of fungus gnats are not very good when you're fighting black root rot because they're very good at moving it around. So you want to have very good fungus gnat control so that the disease stays where it is and doesn't move further. And definitely avoid having reused flats or pots that you've not bothered to disinfest. Those will set you up for a disaster, especially if you're taking the old pansy flats from last year and using them again this year. Be especially careful with the plants that are highly prone to black root rot. Your IPM methods to cope with it involve not reusing those. You can use various disinfectants. There's data that Xerotol works against it, bleach works against it, also Physan 20. So if you get rid of the particles and then treat with a disinfectant after that, you'll have a good program. You want to keep your pH ideally more like 5.5 for the crop, well below 6.0. Have good drainage in your mix, manage your fungus gnats, inspect your plugs. And then when you're working with chemical control, it's very good to be using some thiophanate methyl. You can find that in Banrot or in 3336, other products as well. That is the most effective ingredient against the disease, but it's a group 1 fungicide that's prone to resistance development. So please do rotate that treatment with some others. And Affirm and TerraGuard Medallion are other materials that are effective against the Thielaviopsis that will help you control the disease. Powdery mildews, another fungus. This one's much less subtle. It's just right there in front of you, white fungus growth on the surface of the plants. It's hard to identify at first, but once it gets growing well, once it's very happy, it gets pretty conspicuous. And dahlia is one of the crops where we see the most problem with it, particularly in recent years, it seems. And it can take on many forms, but it tends to spread across the leaf surface rather than make just a few little round spots. So that you'll have, in some cases, just a grayish look to the foliage if the powdery mildew has spread completely across the leaf surface. There's actually the possibility of more than one powdery mildew, just so that you know. We can call them all powdery mildews, and we can treat them all pretty much the same way because of their similar susceptibilities to different chemistries, for example, or to different environmental features. But there can be a lot of different organisms that might affect a dahlia, for example. In this case, it's most likely going to be Golovinomyces chicoracearum, but the others are possible. The only time it would really matter to you would be when you have other plants that seem to have caught the same mildew and you want to know if that's possible. So we know information about the host range of the different powdery mildews. So there might be once in your lifetime a reason why you'd need to identify which one you were coping with. You might find you have problems on poinsettia with the powdery mildew. And you can see here that the powdery mildew of the poinsettia is going to be able to go after bracts as well as leaves. From the powdery mildew's point of view, that fungus thinks that a bract is just as good as a leaf. And that's unfortunate for us because that's the part we want to look attractive. Even if it is free snow, it still is going to make those plants unsaleable. With hydrangeas, you will see it on the bracts as well. They have bracts too, but usually we're complaining more about the disease as it presents on the foliage of the plants. Another one of the crops that's more prone to powdery mildew is verbena. And this has improved a great deal in recent years. For a long time, it seemed like there was very high susceptibility in some of the verbenas that we were growing. And now I think the companies that produce this plant have gotten rid of the most susceptible ones, and we're only rarely going to find a plant that's extremely prone to powdery mildew. The trick with the verbenas is that they can be infected, but it not be very obvious to you. So do ask the question. If your verbenas look funny, look very closely. There's again a long list of possible powdery mildews on verbena, but the one that I see the most often is the last one listed there, the Podosphaera xanthii, which has another handful of plants that you might grow, which could be other hosts for the disease. The trick is that often it's not very white. It can make the plants look just yellowish or purplish. There could be powdery mildew growing on the undersurface of the leaf, for example, and the top of the leaf might just look yellow, and you might feel like adding fertilizer. So just recall that you need to look around and see if you see any of that characteristic white powdery look of a powdery mildew when you see what you think is nutrient deficiency. Here you see more of that purpling, but there's white on top of it. And if you can't see it with your naked eye, a hand lens would help you see that. One of the things that we've noticed a lot in hanging baskets, whether they're down on the bench or hung up already, is that the lower foliage may die. So again, if you see lower foliage dying on verbenas, don't think that it's necessarily drought stress. Look and see if there's a powdery mildew there, because until you address the powdery mildew, you're just going to have more and more dead leaves, and there's more and more inoculum being produced. The way that the susceptibility seems to go with this disease on this plant is that you can't just go by the series. I've noticed in many cases that one member of a series, in this case, the lanai peach, might be very prone to the disease, while other members of the same series might not have any susceptibility at all. So if you decide that a plant doesn't grow well, maybe you just don't want to grow that particular member of a series. You won't necessarily have to drop the entire series. One funny thing about the disease on the verbena is that it's actually the same fungus that is attacking cucurbit crops out there right now. The last vestiges of the pumpkin crop have this particular powdery mildew on them. So if for some reason you're growing something like zucchinis in your greenhouse at the same time that you're growing verbenas, the verbena could be a source of powdery mildew for the squash and vice versa. So that might be a helpful thing to keep in mind. We do fortunately have a lot of fungicides that can be used effectively against powdery mildews. You just need to make the identification before the disease gets very entrenched. And if you're doing good scouting, that will be easy for you. You'll notice in yellow here the groups that are the FRAC groups, the fungicide resistance action committee groups that help you know how different the active ingredients are. And different numbers are important for allowing you to truly rotate rather than putting the same kind of effect over and over again against a particular fungus. The best materials against the powdery mildews, in my opinion, are the strobilurins. In particular, things like Insignia and Compass and Pageant seem to be especially effective against them. You also can find this group 11 chemistry in the 7+11 materials now, such as the orchestra and the broad form. Then group 3 also has some very good effect against powdery mildew, which gives you the TerraGuard and Bannermax type materials, the Bannermax only being used outdoors, the TerraGuard being usable in the greenhouse. We have the new materials Cydo. You also have copper materials, sulfur, potassium bicarbonate, and then your biological controls will work very nicely against powdery mildew. So you can use them either alone at a weekly interval, or you can use them in alternation with one of the chemical materials to help reduce the number of times you need to use a chemical fungicide. And then there are also botanical materials that are good at quelling powdery mildew as well, things like Regime and Regalia and EcoSwing. So you've got a lot of options, and you just need to work out a good program for any of your crops that are prone to this particular kind of problem. The thing that we're always telling you is that there could be a problem with resistance development. And I just wanted to give you one specific example here with that Potosphaera xanthii that goes after the verbenas. There is a record of resistance that's developed to the group 3, group 11, group 7, and group 1 fungicides. That doesn't mean that the one that you have in your greenhouse is necessarily resistant to those. In fact, it would be very surprising if it were, but it means that somebody somewhere on maybe a cucumber crop growing in Israel or something made a report that they saw resistance. By rotating, we slow the whole process of resistance development, and we maintain a longer time period where that fungicide is useful to us and the rest of the industry. So definitely work with rotations when you're trying to control powdery mildew. There are 2 groups of materials that are available to you for powdery mildew control. Just to help you organize your head on this a little bit, there are some that are systemic. These are the ones that are prone to resistance development, but they're also extremely effective. So you want to use them, you just don't want to abuse them by using them overly repeatedly, and you need to rotate them with other things. So here's 3 of your systemic groups that you might use, and then you can easily rotate these with contact materials, such as the coppers or sulfur or millstop, or the biologicals or the botanicals. These don't go inside the plant. They work in a different way, and their mechanism is so different that it really does create a good rotational program when you can alternate them with the systemic materials. Okay, another fungus, Fusarium. This is responsible not for leaf spots so much as for things affecting the roots or the stem or getting into the vascular system and wilting a plant. There seem to be more Fusarium diseases all the time. I think I had 8 or so cases this summer. Here's one that you can run into occasionally when you grow hostas. You can see that root system doesn't look exactly perfect and isn't very large. And above you have scorching of the foliage. That type of look might indicate Fusarium that started as a root rot and moved into the vascular system of hosta. Here's an Echeveria, one of our newer crops, seems to be quite prone to Fusarium disease. And that can cause rotting of the lower foliage. In this case, it's a little wedge of foliage on one side of the plant. Somewhat associated with conditions. You certainly don't want overly wet conditions for this crop, but it's also about sanitation and about what you're dealt. So find yourself a supplier that doesn't sell you Fusarium along with the Echeveria. Another crop you might have had trouble with regard to Fusarium is begonia. The Rieger begonia crop in particular is prone to Fusarium Impatiens, which was originally in Europe, but then was shared with us. And you will see reddening or yellowing of portions of the foliage when this disease is operating. It can be mistaken for a lot of other things, but with time it starts getting more and more obvious, and in a laboratory they can isolate the pathogen. There's essentially a stem rot in this case. So when you just let it sit there long enough, you're going to have brown rotted areas of stem tissue. The most commonly seen disease in this category is on chrysanthemums. The Fusarium wilt disease of mums is unfortunately common, but not usually that devastating, because in my experience, at least, we tend to see it on 1 or 2 cultivars in a season. It doesn't seem to go across cultivars very well. It probably is often representing a slight slip in sanitation back at the source that allowed one cultivar to be not perfectly clean. And fortunately, the industry is always working against that problem. Always report it, because they need to know there's a problem if you run into Fusarium wilt. It does take a laboratory diagnosis to know whether you've got wilting from Pythium root rot or wilting from Fusarium. And there are also stem rot Fusariums, as well as true vascular wilts that are more of a xylem infection. And then at times you might run into this same sort of wilt type disease on crops like gladiolus, which will also get very scorched. That's its version of wilting, I guess. When you're working with Fusarium control, there are certain fungicides that are appropriate. The strobilurin group, again, is useful. Heritage is one of the ones that's used against Fusarium with some effect. Thiophanate methyl helps a little bit. Medallion and Terragard are a little stronger than the thiophanate methyl. But don't think of Fusarium as a disease that you can actually control chemically. You need to have really good sanitation. You need clean stock. You don't want to rely on fungicide chemistry, because it never gives you 100% control. It's more like a 50% control. So it's not sufficient, and we need to have a really integrated program to solve the problem. From research that was done years ago on chrysanthemum, we know that things like calcium levels are very important. So you want good high calcium levels in your soil. When you can, use nitrate nitrogen rather than ammonium nitrogen, because the nitrate nitrogen does not foster the growth of the Fusarium. It's a more expensive fertilizer, but it's very good in terms of management on crops that are prone to the disease. You want to keep your pH high. Notice that this is the reverse of the Thielaviopsis black root rot. If you want to avoid Fusarium wilt, you want a high pH, over 6.2. You don't want to over-fertilize, because nitrogen sort of feeds into the Fusarium fungus. And you want to use some biocontrols at times and the appropriate fungicides to try to curb the Fusarium a bit, and certainly use resistant varieties. If you're growing a crop, and I'll just mention mums as an example because it's fall, and you see a problem in a particular cultivar, that doesn't just mean that it has a problem this year. It means that that particular cultivar is prone to the disease. So it's good to quit growing that cultivar and seek out ones that have the same color, that don't have that high Fusarium susceptibility. Okay, another fungus. I think we're seeing a little bit more in the Cercospora leaf spot category than we used to. Once we started using benlate as a fungicide, these leaf spot diseases almost disappeared for a while, but they're sneaking back in a little bit. So stay vigilant. One crop where 20, 30 years ago, we used to see spotting all the time was pansies and violas. I think that problem has been cleaned up to a large extent, but it's one of the things that might be involved if you see spotting on pansies or violas. Here's a disease I've seen more recently. This is hydrangea with its Cercospora leaf spot. The spots tend to have more of a tan center and a purplish border to them. And the spores are really large and long. They splash very easily from place to place. So if you think you're seeing this, try to get management going quickly before it moves around a lot. A more recent disease that we've only seen for a couple of years, and actually not any, I think, last year, but the year before and the year before that, is a Cercospora leaf spot on begonia. And these spots were fairly large, dark brown spots with a very distinct yellow halo. So it was a fairly distinctive thing, and surprised us, because we weren't used to leaf spot diseases on begonia. We were more used to powdery mildew on begonia. But if you run into this, you'll need to address it by throwing away the symptomatic plants and protecting the rest with fungicides. Some of the fungicides that work against Cercospora, one of these leaf spotting fungi, include things like Daconil and Protect that you might be using fairly commonly in your greenhouse. Thiophanate methyl also works against it unless it's become resistant to that chemistry. The strobilurins can also work. and also things like the tridimethon, the balaton, or if you're outdoors, a Banner Max type of material. And the other side of things is to manage your environment for the plants. Certainly when you're trying to control a fungal leaf spot, you really want to keep the leaf wetness down. So managing the humidity in the greenhouse to keep leaf wetness from being extreme, paying a lot of attention to times when the temperature drops in the greenhouse that tends to create a wet leaf surface, watering early in the day so surfaces dry out before nightfall. All of those kinds of things allow a leaf spot problem to be small rather than large. So if you see a little of it, make sure that you're addressing perhaps a little bit of a shift in your management techniques, because cultural management can be really very effective against these kinds of diseases. Okay, let's move from the fungi to the bacteria. And again, when you're trying to manage bacteria, you need to think about water. You also need to think about seeds, in particular with some of the bacterial diseases, and you need to think about clean stock production, because that's what's between us and some of the worst of the bacterial diseases. And I'm going to focus today on Xanthomonas because it's a relatively frequent plague in our greenhouses, and it seems to be coming up a lot recently. In particular, on the geranium crop this year, we found that growers who received offshore cuttings, even directly from offshore, were running into symptoms like this in propagation. They were seeing wilting, yellowing, and stunting of plants. It's hard to really pick out things that are extremely diagnostic for the disease in question, especially on cuttings that have just been shipped, because you'll notice, for example, at the right, there's what looks like a tear in the leaf, but there's also something that looks a little bit suspicious in terms of yellowing. And then you combine that to the fact that there's some spots over here more to the left that are tiny little brown spots with a bit of yellowing where there isn't any visible wound. And you start thinking, gee, maybe I have a possibility of the bacterial blight disease on geraniums, which is exactly what's going on here. The combination of wilting symptoms along with leaf spots is a very good way to make a field diagnosis of that problem. And fortunately, growers with experience were easily able to realize that there was a chance of bacterial blight this year. And they were able to throw plants away that showed the symptoms and grow the rest of their crop in peace in many instances. One thing that made this year's outbreak easier to cope with was that we now are able to work with immunostrips that are produced by the Agdia company to do a test for either Xanthomonas or for Xanthomonas hortorum, which is the specific one you care about here. And if you've not used these strips, I hope you'll never have a reason to use them, but it's really very helpful to get a quick read on a symptom that you're curious about. And as with a COVID test, very similar technology, you will have one line on a strip if the test is working, and you will have two lines on a test if the organism you're checking for is actually present. And you just basically add a quarter-sized piece of tissue to a bag and rub it to release the plant sap, and then insert this little strip and wait about 15 minutes, and you know whether you have a problem or not. This is quicker than culturing, certainly. We were able to also culture out the pathogen from plants that showed symptoms of discoloration. V-shaped wedges were very common, particularly at the base of leaves on some of the zonal cultivars. Yellowing was common. Individual little leaf spots was common, especially on ivy geraniums. And a lot of people managed to forget that ivy geraniums were also susceptible, and that's a very dangerous thing to forget. The Pelargonium peltatum are just as susceptible as the Pelargonium hortorum. Sometimes the cultivars showed reddening, which is a little unusual, might make you think you've had a phosphorus deficiency, but when you see it along with little tiny round brown spots, that's when you have to worry. And here you see, again, two positive strip tests for that kind of situation. This is a sample that was received in Maryland, and it shows you why we really have to pay attention to this disease. It really destroys the crop quality. So you have one healthy plant here, and beside it, one that's very stunted, where all the lower foliage is browning out. And given the temperatures of summertime, this plant would die completely. So we do need to cope with it very effectively at the greenhouse level and not let it get out to customers, because it will be a very, very bad thing for one's reputation. Excuse me. Culturing method is very important for making sure that you have this disease rather than another one. You can't really trust just an immunostrip test. And we also do inoculation into cuttings, because when you stab a toothpick into a stem and then add a little bacteria into that toothpick wound, it only takes about a week before you get a little canker in the stem when the Xanthomonas hortorum pathovar pelargoni is present. So you can imagine from this story that it's extremely important to scout in the greenhouse. If you're not watching your geranium crop, this kind of thing can fester and get widespread rather than being detected early and thrown away in a way that preserves a lot of your cultivars or perhaps your other sources of geranium. This is why we have culture indexing. Diseases such as the Ralstonia disease that we had trouble with more recently, and the Xanthomonas disease. Because they're systemic, you need a laboratory process making the plant safe before cuttings are sent to you. And you need that to have a lot of checks and balances and safety features in it. This is a picture from the offshore facility of Duman, where they have very good signage and a lot of training for their workers. And they're doing everything they can to keep pathogens such as these bacteria from getting into the crop. There is a lot of information provided to the workers so that they arrive clean and they put on gloves and protective aprons as they enter the facility. And they do a lot of things to keep it fail-safe too. There's a tendency in some of these facilities to even hire one employee to check the other employees to make sure that no one's cutting corners. And that seems like a luxury thing, but it actually is the kind of thing that helps keep geranium safe for you. Back a few years ago, people were just washing their hands, and now they're washing all the way up to the elbows to try to make sure they're not introducing the pathogen into a facility. So when you're thinking about cultural controls for Xanthomonas, they're very important because the disease will occasionally slip through production, and we need to be really vigilant to make sure we're not letting it take out entire crops for the final grower. You need to have those culture index cuttings, or you can grow just hybrid seed geraniums. They've not had a problem with Xanthomonas other than catching it from the cuttings. Sanitation is really important. Scouting is important. You don't want splashing. And then there's some rules here that we were very familiar with back in like the 1980s that I think a lot of people had forgotten by the time we got to the 2020s. So it's not smart to grow ivy geraniums, the Pelargonium peltatum, vertically over zonal geraniums at a bench level. That allows sick ivy geraniums to drop bacteria onto the zonals. And this happened this year. I was very sorry to see that happening. It's also important to keep geraniums grown from seed completely separate from ones that are grown vegetatively, because the seed geraniums are going to be fine if they're not brought into contact with vegetatively propagated geraniums. It's also important to keep sources separate. And that doesn't mean the same company. That means if it comes from the same cultivar, even grown on different continents, might have the same problem. So you want to think more about a greenhouse propagation center as a separate unit and keep those separate in your greenhouse, in separate structures if possible, separate benches if not. Any separation you can do will help you minimize losses when a problem like Xanthomonas comes through. And then absolutely avoid ebb flood irrigation or capillary mats or anything that's a water flow that can go from plant to plant, because the bacteria, just like those Thielaviopsis spores, are able to easily move with the flow of water. And that's a serious level of spread. There was one interesting trick that was developed by Steve Namath at Ohio State in 1994 that people have asked about again this year. And that is that if you work with the material called Phyton 27, and I'm not sure if this applies to Phyton 35, but someone should check it out. You can essentially stress the plant a little bit. And if it's already stressed by Xanthomonas infection that's systemic, the plant may turn yellowish, and then you can pick it out. And this trick only works out when you have warm temperatures, 80 degrees Fahrenheit or warmer. If it's February and your plants are much colder than that, you're not going to get any benefit of the treatment. And it doesn't cure the plant or anything like that. It just makes the ones with the infection look different from the ones nearby that don't have the infection. So it can help you with your roguing out when you're trying to control an epidemic. We have tried doing chemical control of this bacterial blight disease, and it does not work, even though there are differences in treated plants. And this is some of Ann Chase's work here, showing that compared to inoculated plants that weren't given any treatment, which had over 80% infection, with Phyton 27 treatment, the level of infection could be brought down. With Rhapsody treatment, which is a Bacillus subtilis, there could also be, with the high rate, the 2% rate, there was a real statistical reduction in how much symptom there was. And with Camelot at a 16-ounce rate, there was also an improvement. And here's some of Dave Norman's work, which shows you a similar thing with over 20% leaf spots per plant in the control where there was no treatment done, and then benefits from Coside and Camelot and Junction and Pencozeb and Cuprofix, all of which have copper and some of which have manganese in them as well. So you can reduce disease, but you just can't trust it because there's always some survival, and you're actually much better off throwing away an area that's contaminated by this pathogen rather than trying to truly pick out individuals. Another Xanthomonas disease caused by bacteria, but a different bacterium once you get to the species level, is the one that goes after cabbage and kale. And this is one that just drives people crazy because it can be seed-borne. So you can start out with it very early in the crop. If you see contamination in a seedling flat, it's a good idea to just get rid of that flat because it's so hard to control a bacterial disease. You can do seed treatments, however, and one of our New Yorkers is known to have been successful for several years now doing a hot water seed treatment to eliminate the bacteria from his cabbage and kale. So seek that solution if you're willing to go to the trouble of doing that seed treatment. There's information on the web supplied by Meg McGrath, who is the pathologist that I work with here at the Long Island Hort Research and Extension Center, that spells out the temperature and the methodology for that. There can also be a seed-borne problem with a Xanthomonas bacterium, a different one again, on zinnia. This is Xanthomonas campestris pathovar zinniae. And it's easy to identify, except for the fact that it looks almost exactly like an alternaria disease, which is a fungus disease. With both of them, you can have these sort of blocky kinds of spots and some yellowing. So if you have a problem with zinnia, it's going to be worth your while to figure out what the agent is. Using copper will help to retard both of those problems, but you could probably manage the alternaria disease pretty effectively if you knew that's what you had. And again, with the bacterial disease, you need to throw plants away because it's so hard to manage bacteria in any really effective way. The disease can get quite extensive, and areas will even fall out from the leaf if there's enough of the infection going on. And we see an amazing amount of this from time to time, again, often associated with one cultivar in a season. If you want to avoid the zinnia disease pretty much altogether, you can work with certain kinds of plants that have incorporated some angustifolia blood in them. So the narrow-leaved zinnias don't have the high susceptibility to the Xanthomonas problem. So if you work with things like the Profusion series, you've got reasonably good resistance to Xanthomonas there, as well as resistance to powdery mildew and alternaria. You have a slightly different look in your zinnias, but you have solved the disease problem. So consider that option if this has been a real headache for you. You can also run into Xanthomonas problems on ivy plants, not necessarily just the English ivy. Absolutely any ivy can get it. And you'll tend to see dark spots with a yellow halo in that case. And you might run into problems if you're trying to use a lot of copper because the foliage can get bleached out a bit by the copper treatment at times. You might find it helpful to use a biocontrol in between copper treatment to reduce the amount of copper that the tissue feels. We also can have bacterial problems in begonia crops. This is a different Xanthomonas. This one is Xanthomonas axonopodus pathovar begoniae. And it's fairly easy to recognize after way too much practice. I've noticed that almost all the time the lesions will start at the edge of the leaf and make a wedge. And almost all the time you'll see sort of a freckling at the leading edge of the lesion. And that's apparent here. A lot of little tiny water soaked spots are occurring out here in the greener tissue, and also can be seen within the brown tissue as the bacteria are moving into the leaf. Eventually, they'll get into the stem on many of the cultivars and species of begonia, and then you'll have a stem rot and a complete plant collapse. So again, you need to respond to this disease by throwing away plants with the symptoms and getting a diagnosis first, so that you're not throwing away plants that just have scorching because of drought or some such thing. Here's another example of the symptoms of the Xanthomonas in the begonia leaf. We've also seen some problems with Xanthomonas recently in the propagation stage of poinsettias. The problems haven't been following through to the final crop very often. Many of them seem to only thrive in the wet situations that you have during the propagation of the disease. But certainly understand what they are. Don't grow any more wetly than you need to. Throw out plants with the problem, and make sure that you don't think that a fungicide is going to solve this. This is actually a bacterial disease. The treatments for bacteria are very limited, which is why I'm saying throw out a lot. Copper is one of the few active ingredients that works, but if a plant has been in horticultural production for many years, it's quite possible there's copper resistance in the bacteria that are bothering that plant. So that means it's just like spraying with water when you spray with a copper material in those cases. It's still, unfortunately, the best thing we've got. And then second to that, you will find benefit from treating with things that contain Bacillus subtilis. That's a biocontrol. It's a bacterium itself, and it is able to compete with the bacteria that cause the disease. And we've had reasonably good benefit from using that treatment either alone or in alternation with copper. The AgriStrep that's listed here, I just am putting in to be thorough, but it has a very narrow label. There are not many things you can use it for that you might be growing in the greenhouse. And it does tend to cause some real injury to the geranium crop, which is one where you would most like to be able to control a Xanthomonas. So don't try it on geraniums, because it'll make the leaves all turn very whitish yellow. Here's a longer list of some of the copper materials that you might work with. All of these have their ideal times to be used. If you want one with less residue, you want to choose one that doesn't have a thick blue coloring to it. This is one of the advantages to using the biocontrols, is they don't give you a blue surface. And you'll get a little benefit from the phosphorus acid materials, but they're not strong competitors with the copper materials, I don't think. Another bacterial disease that we've seen a bit of again this year, unfortunately, is a very old classic one that's been studied since 1901, I think. These are argyranthemums, or argy-bargy, as they call them in Great Britain, with the crown gall disease caused by Agrobacterium tumefaciens, a particular bacterium that essentially mates with the plant cells and transfers genetic material into them, does its own little genetic engineering experiment. And then as a result, you'll see swellings at the root crown, which is where the name crown gall comes from, or possibly further up the plant, because anytime there is a wound, it allows an expression of this gall formation that you're looking at here. This is what we've seen.
Margery?
Yes.
For a moment, can we do a pause for a poll here?
Sure,
We're over the halfway point. I know you put a slide in somewhere, but
go ahead, do a poll.
JC, are you able to get that up? There we go. Look at you all clicking away.
Oh, there are people out there. That's reassuring. I can only see 4 people, but I'm just taking on faith that there's more out there. And indeed, it looks like there are 84 out there.
At least.
And there are only 6 of them that are asleep, but maybe some of those are the organizers.
Well, you were talking about treatments for bacteria. Someone asked about double nickel.
Oh, yes, that's another one that's in that category of, it was originally started out as a vegetable treatment, but it's a possibility for some of our other greenhouse crops too. Yep, that one's in that effective category for sure.
Yes.
Thanks, Rosa. I can't see the questions at all. So.
All right, it's been a minute, so we can end the poll. And we're good. All right, carry on, Margery.
Thank you. I wasn't sure how important the halfway point was. I was mostly just trying to.
We like to keep it random to keep people on their toes.
Okay.
And we do try to wrap up by 1 because
I'm watching.
in respect with other people's time. All right.
Right.
Thank you so much. Carry on.
Okay, I'm just trying to get rid of that little pole thing. There we go. I figured I'd have to get rid of it. Okay, so again, crown gall can cause these little tiny galls anywhere on the plant. And we've been seeing symptoms like this on Shasta daisy this year and suspecting that it's a crown gall problem, but the diagnosis is currently going on, so I can't swear to it right now. And then there's another symptom we're seeing on dahlias, which appears to be the leafy gall disease. Again, the diagnosis is currently underway. But there is an organism that lives primarily on the surface of the plant, but also goes inside, which knows how to do things like put out a lot of cytokinin and get a lot of shoots produced. And it makes some pretty ugly root systems. So if you have strange symptoms like this, you do need a lab to help you figure out if you're facing either crown gall or rhodococcus. And again, this is a disease that's dealt with by throwing plants away. See how close you were to show poll? So, all right, good that we have that done. So nematodes, this is a category of pathogen where, again, you need to think about water and you need to think about clean stock because you don't want nematodes in your production. Nematodes are actually worms. And I've had an old friend has been arguing with me recently, like, how can a worm be a pathogen? Well, a worm can be a pathogen because when it interacts with a plant, it creates symptoms that we call a disease. And there are only certain ones that have that ability. But this particular one has a beautiful little mouth part called a stylet that looks like a little needle here at the left end, and it pierces into the plant tissue and feeds on it and acts as a parasite. And you can see how tiny it is. They're only a 64th of an inch long, sometimes up to maybe a 16th of an inch long, maybe about a millimeter. And large numbers of them can have real impact on plants. Mostly in the greenhouse world, we're only going to see foliar nematodes because we're growing in soilless mix. And that's really freed us up from a lot of nematode problems. But in Bergania, you might see these kind of wedge areas. And 2 other crops to be really alert in are Japanese anemone and echinacea, especially if there's overhead watering, you're going to have a chance of running into foliar nematode symptoms. And again, throwing this away is really important. In the virus category, the water isn't so important anymore. You really want to think more about the clean stock idea, how you're handling the plants and the things that might be vectors, carriers of the viruses. We have a large number of viruses that we might encounter, and there are more being discovered every day. And I know that because I've helped to identify some new ones. We have such good molecular genetic techniques now that it's easier to figure out that you have a new virus problem. The names of viruses don't tell you everything that a virus can do. They only tell you one of the things a virus can do. So INSV, for example, can infect impatiens and can cause necrotic or dead spots on an impatiens, but they can also do a lot of other things like stunt plants and curl the leaves and do this and do that. So you just have a name as a handle for the organism. Don't think it's telling you the whole story. And you might encounter viruses that you've never heard of before and that no one's ever heard of before. That's the way things went this year, for example. One of the news items was the chili pepper mild mottle virus. And that's one that no one had heard of at all until about a year before when it was seen on peppers in a field in Peru. And yet all of a sudden we had it in our greenhouses. And that's a virus where spread can happen just from handling. So keep that possibility in mind when you see virus symptoms. Unless you know it well, there's a chance that you might just be able to move it by handling. So don't touch a funny symptom and then go touch the rest of your crop. If you're a touchy-feely grower, you might end up with virus problems. In the case of something like INSV or TSWV, the Impatiens necrotic spot virus, or the tomato spotted wilt virus, you know that thrips are going to be able to spread the virus. So thrips management is the most important thing you can do for managing those diseases. In some cases, the virus symptoms have been considered positive. In the 1600s in Holland, they were actually spending huge sums of money for tulip bulbs that they thought would give them these kinds of patterns in the petals. And it turned out later on that this was actually a virus tulip flower break virus, and the plants would die after a year or two because they had a virus in them. So these were not good investments, and I would not suggest that any of you invest in viruses either. A number of things that you might run into from time to time. Here's tobacco rattle virus. This one is actually spread by nematodes. Here you've got rose rosette virus. That's a newer one that's very problematic for us now in nursery production. It's actually spread by these little tiny microscopic Aphelenchoides mites called Phyllocoptes. And you can't even see these unless you're very good when you're using a microscope. They're hiding under some of the plant tissue and really teensy. They're pretty ugly too. They sort of give me the shivers when I do find them. But this is the way this particular virus moves around, and we didn't even realize it was a virus until a few years ago. The symptoms are going to include strange amounts of reddening on the plant tissue, what we call hyperthorniness, more thorns than you would expect on that particular kind of rose, and also just a continued reddening of shoots. Your new shoots might ordinarily be red on a rose, but they don't quit being red. That's a very odd symptom of this disease. There's also witch's brooming. And the main thing I wanted to mention to you is that the multiflora rose, which is basically a weed for us in the Northeast, is one of the places where this virus can hang out. So if you want to be a rose producer, you want to first get rid of all the multiflora rose around the edges of your property, because the little Aphelenchoides mites there might be able to waft over in a breeze and come over to your crop and spread this particular disease. It's not uncommon in multiflora rose in the Northeast. The tomato spotted wilt virus is pretty common to you now. It's not super common in greenhouses anymore, but it's a possibility, a real possibility. We see several cases every year. Pericallus is one of the crops that's been troubled by it recently. We've seen a few cases in chrysanthemum getting into the stems, causing very different kinds of symptoms. We know that the thrips larvae of the western flower thrips will pick up this virus. And then when the thrips is an adult and has wings, it's able to spread the virus off to more plants in the greenhouse. So that thrips management is extremely critical for you. You can have crops like lobelia that are already in hanging baskets, quietly getting these tan symptoms in the foliage that are an indication of TSWV, because thrips can find them just fine. You just can't see the plants too well because they're over your head. Again, we can use these immunostrips or any other test kit that you have that's a virus identifier. Sometimes we want to know whether it's INSV or TSWV. Mostly from your perspective, you don't care. In this instance, you're looking at a TSWV case causing these blackenings on the impatiens stem. And you can see that it's not INSV because there's no secondary band here in that instance. Most of the time, you're not going to care because either of those TOSPO viruses is going to mean that you need to throw the affected plants away and get very, very good thrips control as soon as you possibly can. Again, here's some of the spotting from the second virus in this category, the Impatiens necrotic spot virus, that tends to cause some sort of black symptom on the foliage. Whoops. And then here's the INSV appearing again on Echeveria. Very curious symptom here with concentric rings on the foliage. Not something that INSV usually does at all, but that's what was in this tissue. And then if you're still growing gloxinias, you're very brave because that crop's terribly susceptible to both thrips and INSV, and it creates all sorts of interesting patterns on the leaves. And then at the right, you see a set of symptoms that would be hard to identify, some browning of leaves, some yellowing in the foliage, and that's from INSV as well. And then if you're getting back to foliage plant culture, which is happening in portions of the Northeast, do keep in mind that some of our foliage plants are also susceptible to INSV. It's a question of whether thrips like to feed on them. And this yellow blotching on this particular philodendron was caused by INSV. We had one interesting case this year where there were symptoms being seen on New Guinea impatiens that could have been botrytis, could have been virus, not really sure. But then when they showed me pictures of what was going on with their begonias, this is a dead giveaway that you've got a tospovirus problem. It looks like the plant is absolutely variegated. The systemic infection within one leaf will cause all these yellowed patterns that are quite intriguing in that crop. So by adding the two things together, you can pretty much guess that you've got a New Guinea impatiens problem with INSV as well. And then in this particular greenhouse, the extension specialist also crawled around on the floor to please me and look for weeds that might be hosts. And he found chickweed. And lo and behold, when we ran tests, we found the chickweed had every bit as much virus inside it as the begonias and the New Guinea impatiens. So do be alert to weeds if you have this outbreak, because they can be a way of keeping the virus in the greenhouse, even if you throw the crop away. And then here's our newbie, the Calibrachoa mottle virus that we saw this year. This was reported for the first time just this September as a new virus. It can cause yellowing and purple mottling and a tip necrosis and color break in the Calibrachoa plants. And we know it can also cause peppers to show symptoms in Peru. So it's good to know two things about a virus, even if we don't know much else. The only other things we know about it are that it's a tobacco virus, meaning that it's in the same family as TMV, which is a relatively well-known virus, the tobacco mosaic virus. We have a lot of fears for TMV getting into our petunia or Calibrachoa production. In this case, we have a relative of TMV, this chili pepper mild mottle virus, getting into Calibrachoa in this spring season. Hopefully it won't come back, but it causes, as it says in the name, only a mild mottle in the foliage. And then we are concerned that it would probably be spread by handling because it's a relative of TMV, which is spread by handling. And we certainly saw that we were getting positive results when we used TMV test strips. Even though it's not TMV, it will give you a positive with a TMV strip. And you can see at the right, really impressive color break symptoms from this virus. And it didn't happen just to the yellows. You could see it in all different colors. And if you look at the left there, you can see that the infected plants are quite stunted. So you'd have the combination of stunting, which is a poor crop quality, along with a real destruction of the normal coloring in the petals. Really not one we want to keep. So I'll leave you with the question of, this should have been your poll question actually, which of these things would you least like to encounter given the chances of fungal problems or bacterial problems or nematodes or viruses? And I think it's a really hard choice. You don't really want any of them. Ina here really favors the fungi, so she would pick fungi every time. And I have my email address there in case any of you don't have time to ask questions today. Feel free to email me with questions. And I would be happy, Rosa, if you have any questions from the chat to try to answer some.
Yes, thank you. We have two questions. You can type your questions if you want to, or unmute yourself. I'm going to read these two in the meantime. The first one is if you can talk a little bit about Xanthomonas on rosemary. So suggestions and management, but perhaps also describe a little bit of the symptoms for the rest of it.
Sure. The Xanthomonas on rosemary is not something that I'd ever read about, but I did encounter it probably 5 years ago. And in the cases that I've seen, and I've only seen a few, the spots are very small and there can be yellowing around the spots. Isn't necessarily yellowing, but that makes it the most easily identified. But you have little tiny brown, often half moons on the edge of the leaves. And I think the main problem is it's hard to see them. It's a fairly dark leaf and people don't always notice that they have the bacterial spots, and then it can get spread by splashing of irrigation. So again, I would watch your rosemary and be very alert to that kind of a symptom and throw away any plants that show it. We have so little we can treat an herb with that I don't think we'll have very good chemical control success. Working with maybe some of the Bacillus subtilis materials that would be legal for use on an herb would be helpful as a precaution, but certainly minimize your overhead watering and certainly watch for symptoms and throw away plants with the problem.
Okay. And then the next question is, do feltine nematodes parasitize plant parasitic nematodes?
Ooh, what a lovely thought. There has been a little experimentation along those lines. So the Steinernema feltii is a beneficial nematode, not an evil nematode. And it's used against insect pathogens. And there is some competition, I think, from what I've read. It's not been found to be a super good control, certainly, but I think there is some interaction. And maybe it helps keep some of them out if they get there first or something along those lines. I keep hoping for a solution along those lines for our new beech leaf disease that's happening in the forests now, where you have nematodes in the foliage of beech trees that are introduced, we believe, recently into this country. And I would love to spray on some counterattack nematode to go after the first ones, because you need something tiny that can get down inside the leaf and go after those. So I don't think it's a practical control to work with the Steiner nema in that fashion, but it does work very well against some of the insect problems.
Yeah, and we have one third question. And they're asking if you can expand on how to apply the Phyton 27 to identify the Xanthomonas and geraniums.
So the only things I can tell you are to work with the high rate, the 50 ounce rate. That's important. The lower rate doesn't work. And to also make sure the temperatures are at least 80 degrees. And when applied across the crop, that might allow you to tell something about which of the plants are infected with the Xanthomonas. I suspect it would also tell you if the plants were infected with something else, because we don't think it's a specific relationship. So for example, if there were Ralstonia, I wouldn't be surprised if that wouldn't also cause the plants to look a little chlorotic following the Phyton 27 treatment. It was never really studied to the level where we understood exactly what was happening, but the interpretation given at the time was that the plant was already stressed by the internal pathogen in its vascular system, and by adding copper, which was also stressful, the plant would tend to yellow, which is a symptom it will give if it has too much copper in it. So it was just a trick. And there might be a time when we can apply it to help us deal with some of these disease problems. But the way things went last year, basically people had to respond quickly and throw away the infected plants, or they had way too much spread. So don't play around with unusual things when you can actually throw the problem away. It's really a good idea to throw it out.
Yes. Well, it's 1 o'clock. Thank you very much, Margery. I think we're going to close for questions. You have Margery's email there. You can always reach out to her. And then I'll hand it up to Sarah, because I think she has something else to do regarding pesticide credits. That's it.
All right, I put the link in the chat for the attendance verification survey, your last one. Thank you all who have been here through the whole series or have popped in and out. That link will also be emailed out following the webinar here by JC or Leslie. So with that, you're welcome to carry on with your day. Thank you so much for joining us.
Wow.
Thanks, Rosa.
Thanks. Bye-bye. Thanks,
All right.
everyone.
You got it, JC. You're going to shut us down.
Yep, just making sure everybody's all set.
I think we're good.
Okay, thank you.
Bye.
Bye.